Two tomato plants stand less than a meter apart in the same garden bed. Same variety, same soil, same sunlight and water. But one struggles with pale yellow-green leaves, a handful of small flowers that drop before forming fruit, and thin stems that bend under their own weight. The other plant tells a completely different story. Deep green foliage, thick stems covered in flower clusters, and small green tomatoes already swelling behind nearly every blossom. The difference did not come from expensive fertilizers or years of gardening experience. It came from a simple mixture prepared in a kitchen container using ingredients most people already have: water, yeast, sugar, and a few hours of patience. Yet thousands of gardeners never see results like this because they apply the mixture at the wrong time, in the wrong way, or without understanding what it actually does inside the soil. Because this is not fertilizer in the conventional sense, it is something older and more interesting. Most gardeners assume that feeding tomato plants means adding nutrients directly to the soil. Nitrogen for leaves, phosphorus for roots, potassium for fruit. The logic seems straightforward. But tomatoes often fail even when nutrients are present in the soil. Leaves turn yellow, flowers form but drop without setting fruit, and growth slows during the exact moment when the plant should be accelerating toward harvest. The problem is rarely absence, it is availability. Nutrients exist in soil as mineral particles, organic residues, and chemical compounds, but plants cannot absorb most of these forms directly. Between the mineral and the root lies an invisible biological network that determines whether nutrients ever reach the plant at all. Bacteria dissolve minerals. Fungi extend microscopic filaments that transport nutrients across distances roots cannot reach alone. Organic acids produced by microorganisms reshape the chemistry of soil particles, releasing elements that would otherwise remain locked. When this biological system functions well, plants thrive even in soil that chemical analysis might call deficient. When the system weakens, plants starve even when surrounded by nutrients. And tomatoes, more than almost any other garden crop, depend on this biological partnership during one specific phase of their life cycle, the moment they begin to flower. Flowering marks a dramatic metabolic shift. Until that point, the plant has focused on building leaves, stems, and roots. Suddenly, it must redirect enormous amounts of energy toward reproduction. Flowers form, pollen develops, fruit begins to swell. Each of these processes requires a surge of specific nutrients delivered at exactly the right moment. Potassium to regulate water movement and strengthen cell walls. Phosphorus to fuel the energy transfers that power flower formation. Calcium to build the structure of developing fruit, and magnesium to maintain the chlorophyll that captures sunlight and converts it into sugars. If any of these elements fail to reach the plant during the narrow window of flowering, the consequences appear quickly. Blossoms drop, fruit set declines, and the plant produces foliage but little harvest. For generations, farmers understood this instinctively. Long before chemical fertilizers existed, they relied on fermented organic materials to support crops through critical growth stages. In post-war Korea during the 1960s and 1970s, a farmer named Cho Han-kyu began systematically studying these traditional methods. His country's agricultural system was rebuilding after devastation, and imported chemical fertilizers were expensive and often unavailable to small farmers. Cho turned his attention to something that had always existed but was rarely examined scientifically: the role of fermentation in agriculture. He observed that in natural ecosystems, nutrients rarely move directly from soil to plant. Instead, they pass through cycles of microbial transformation. Leaves fell and decomposed, fungi broke down wood, bacteria metabolized organic matter and released nutrients in forms that plant roots could absorb. Fermentation, Cho realized, was not just a method for preserving food. It was one of nature's fundamental processes for making nutrients available to living systems. He began experimenting with ways to capture and concentrate this process. Farmers collected indigenous microorganisms from forest soils, fermented them with plant materials and sugars, and applied the resulting liquids to their fields. The results were consistent enough that the practice spread. What became known as Korean natural farming was built on a simple principle: feed the soil biology, and the soil biology will feed the plant. Western agriculture moved in the opposite direction during the same period. Synthetic fertilizers offered speed, uniformity, and the ability to deliver precise nutrient ratios across vast acreages. The biological pathway was bypassed entirely. Nutrients were engineered to dissolve immediately in soil water, allowing roots to absorb them without microbial assistance. For large-scale industrial farming, this approach solved many problems, but it also created a quiet dependency. Soils managed purely through synthetic inputs often experienced declining microbial diversity over time. Organic matter decreased, soil structure weakened, and plants became less able to access nutrients through biological pathways, requiring ever-increasing chemical applications to maintain yields. Home gardeners inherited this same approach. Tomato plants were fed with soluble fertilizers designed to deliver nutrients instantly, and for a while, it worked. But many gardeners eventually noticed something troubling. Their soil seemed to need more input every season just to produce the same results. The biological system that once regulated nutrient flow had quietly diminished. Which brings us back to the simple fermented mixture. Because what it does is not add nutrients, it reawakens the biological system that delivers them. The fermented mixture begins with ingredients so ordinary that most people overlook their potential entirely. A liter of clean water, 1 or 2 tablespoons of dry yeast, and a tablespoon of sugar mixed together in a container and left to sit for several hours. Some gardeners add a small amount of buttermilk or plain yogurt to introduce lactic acid bacteria, though the basic formula works without it. Within 2 to 4 hours, the liquid begins to change. Bubbles form on the surface as microorganisms multiply rapidly, consuming the sugar and releasing carbon dioxide. The smell shifts from sweet to slightly sour, signaling that fermentation has begun. What makes this mixture effective is not any single ingredient, but rather the biological transformation that occurs when they combine. Yeast cells contain concentrated amounts of B vitamins, including thiamine, riboflavin, and niacin, along with amino acids and growth-promoting compounds. When yeast ferments in the presence of sugar, these compounds are released into the liquid along with organic acids produced during microbial metabolism. If lactic acid bacteria are present from buttermilk or yogurt, they contribute additional organic acids that bind with mineral ions in the soil through a process chemists call chelation. Many essential plant nutrients exist in soil as mineral particles that roots cannot absorb directly. Calcium might be present as limestone fragments, potassium locked inside clay particles, magnesium bound to organic matter. These minerals must first be dissolved and converted into ionic forms before plant roots can take them up. Organic acids produced during fermentation help facilitate this conversion by interacting with mineral surfaces and gradually releasing nutrients into the soil solution where roots can access them. When the fermented liquid is diluted heavily with water and applied to the soil around tomato plants, it introduces millions of living microorganisms directly into the rhizosphere, the narrow zone of soil surrounding plant roots where most nutrient exchange occurs. The diluted ferment provides both living organisms and the organic compounds they need to remain active, effectively jump-starting biological processes that may have slowed or stalled. The response begins at the root surface itself. Root hairs begin expanding more rapidly when microbial activity increases around them because certain bacteria produce plant hormones as metabolic byproducts, compounds that stimulate cell division and elongation in root tissue. At the same time, microbial enzymes begin breaking down organic matter in the surrounding soil, releasing nutrients that were previously bound in forms plants could not use. The effect is often visible within a week or two. Leaves that were pale yellow-green deepen to a richer color as chlorophyll production increases. Stems thicken as calcium strengthens cell walls throughout the plant's vascular system. Most importantly for fruiting crops like tomatoes, flower clusters become more abundant, and the percentage of flowers that successfully set fruit increases noticeably. This happens because potassium, the element most critical for moving sugars from leaves to developing fruit, circulates more efficiently through the plant when soil biology is active. Understanding why tomatoes respond so dramatically requires looking closely at what happens inside the plant during the transition from vegetative growth to reproduction. For the first several weeks after transplanting, A tomato seedling directs nearly all of its energy toward building structural mass. During this phase, nitrogen is the most critical nutrient because it forms the backbone of proteins, enzymes, and chlorophyll molecules. But when the plant reaches a certain size and maturity, its metabolic priorities shift abruptly. Flower buds begin forming, and suddenly the plant must allocate resources toward an entirely different set of biological processes. Producing flowers requires enormous amounts of phosphorus, the element that fuels energy transfer within cells. Without adequate phosphorus, flower buds form slowly or abort before opening, and poor phosphorus availability often results in weak pollen that fails to fertilize properly, leading to blossoms that drop without setting fruit. Once pollination succeeds and tiny green tomatoes begin swelling behind the flowers, the plant's demand for potassium increases dramatically. Potassium regulates the opening and closing of stomata and governs the movement of sugars produced during photosynthesis, directing them from leaves through the plant's vascular system to developing fruit. When potassium is limited, sugars accumulate in leaves rather than moving to where they are needed most, and fruit development slows. Calcium strengthens cell walls throughout the plant, but its importance becomes most visible in developing fruit. Tomatoes that do not receive adequate calcium during the early stages of fruit growth often develop blossom end rot, a condition where the bottom of the fruit turns black and leathery as cells collapse due to weak structural integrity. The challenge for gardeners is that all of these nutrients must be available simultaneously during a relatively narrow window of time. Chemical fertilizers can deliver these nutrients in precise ratios, and when applied correctly, they work effectively. But they bypass the biological systems that regulate nutrient flow in natural ecosystems. And over time, this can create soils that function well only when continuously supplemented with synthetic inputs. The fermented mixture operates through a different pathway entirely, one that strengthens rather than replaces the microbial networks that have always mediated the relationship between soil minerals and plant roots. The gradual disappearance of fermentation-based soil amendments from mainstream agriculture emerged from a series of practical choices made during a period when farming itself was being fundamentally redesigned around industrial principles. During the early and mid-20th century, agriculture faced enormous pressure to increase production rapidly. This transformation required inputs that could be standardized, stored indefinitely, transported across long distances, and applied uniformly across vast fields using machinery. Fermented biological preparations did not fit that model well. Synthetic fertilizers offered something entirely different. They could be manufactured in factories to exact specifications, bagged in uniform quantities, and stored for months or years without degradation. Agricultural extension services, university research programs, and government agencies increasingly focus their recommendations around these products because they fit the economic and logistical structure of modern farming. This approach worked remarkably well in many contexts, and it would be inaccurate to suggest that synthetic fertilizers were inherently harmful or that their adoption was a mistake. They helped feed billions of people and supported the expansion of global food production during a critical period in human history. But the system also created certain dependencies that became more apparent over time. Soils managed exclusively through synthetic inputs often experienced gradual declines in organic matter content, which weakened soil structure and reduced water-holding capacity. Home gardeners inherited this same framework. Tomato plants were fed with water-soluble fertilizers designed to deliver nutrients immediately, and the results were often excellent in the short term. But many gardeners eventually noticed that their soil seemed to require more intervention each season, and that plants became more sensitive to stress, disease, and nutrient imbalances. In recent years, a growing number of home gardeners have begun questioning whether the chemical-intensive approach that dominates commercial agriculture makes sense at the smaller scale of backyard vegetable production. Many gardeners notice that their tomato plants seem increasingly fragile despite regular fertilization. Blossom drop becomes more frequent during temperature fluctuations. Fruit set declines even when flowers appear abundant, and leaves show deficiency symptoms that do not respond predictably to additional fertilizer applications. The problem often traces back to soil biology. In raised beds, container gardens, and plots that have been cultivated intensively for years using only synthetic fertilizers, microbial populations can become severely depleted. What remains is a growing medium that functions more like hydroponic substrate than true soil, capable of holding nutrients but lacking the biological machinery to regulate their availability. Fermented mixtures offer a relatively simple way to begin rebuilding these biological relationships. They do not require expensive equipment, specialized knowledge, or significant time investment. The ingredients are inexpensive and widely available, and the preparation process takes only a few hours. The preparation process is straightforward. Begin with 1 liter of clean water at room temperature. Add 1 to 2 tablespoons of dry yeast and 1 tablespoon of white or brown sugar. Some gardeners include a few tablespoons of plain yogurt or buttermilk to introduce lactic acid bacteria, though this addition is optional. Stir the mixture thoroughly until the yeast and sugar dissolve completely, then cover the container loosely to allow gases to escape. Within an hour or two, small bubbles will begin forming on the surface as yeast cells consume sugar and release carbon dioxide. After 2 to 4 hours, the mixture is ready to use. Dilution is critical. Mix 1 part of the fermented liquid with 10 parts clean water, creating a dilute solution that introduces microorganisms and organic acids without overwhelming the soil system. Apply the diluted mixture directly to the soil around the base of each plant, saturating the root zone where microbial activity matters most. Avoid spraying it on leaves or flowers, as the sugars and organic compounds in the liquid can attract insects or promote fungal growth on plant surfaces. Timing determines how effectively plants respond. The ideal moment to begin applications is when the first flower clusters appear on tomato plants. This signals that the plant has entered its reproductive phase and nutrient demand is about to increase sharply. Repeat applications every 10 to 14 days throughout the flowering and fruiting period to maintain consistent microbial activity in the root zone. Several important cautions apply. The fermented mixture is a supplement, not a complete fertilizer. It does not contain sufficient quantities of nitrogen, phosphorus, or potassium to meet all of a tomato plant's nutritional needs. Soil must still receive organic matter through compost, mulch, or other amendments. Plants that are severely stressed, diseased, or damaged should not receive fermented applications until their health improves. When these guidelines are followed carefully, most gardeners notice visible changes within 1 to 2 weeks of the first application. The first sign that the fermented mixture is working usually appears in the leaves. Within 7 to 10 days after the initial application, foliage that had been pale yellow-green or showing signs of chlorosis begins shifting toward a deeper, richer green color. This change reflects increased chlorophyll production as magnesium becomes more available to the plant through improved microbial activity in the root zone. Stem thickness often increases noticeably during the same period. This happens because calcium, which strengthens cell walls throughout the plant's vascular system, moves more efficiently from soil to plant tissue when organic acids help keep it soluble and available. Stronger stems support heavier fruit loads without bending or breaking, and they transport water and nutrients more effectively between roots and leaves. The most significant change appears in flowering and fruit set. Tomato plants treated with fermented inputs during the early flowering stage typically produce more flower clusters per branch, and a higher percentage of those flowers successfully develop into fruit rather than dropping prematurely. This improvement occurs because potassium circulates more readily through the plant when soil microorganisms are actively processing organic matter and releasing nutrients in plant-available forms. Fruit size and ripening patterns also show measurable differences. Tomatoes on plants receiving fermented applications tend to develop more uniformly, with fewer instances of blossom end rot or uneven ripening. This consistency reflects better calcium distribution throughout developing fruit and more stable water uptake through healthier root systems. Root development, though invisible without excavating plants, undergoes equally important changes. Increased microbial activity in the rhizosphere stimulates root hair formation, dramatically expanding the surface area available for nutrient and water absorption. A plant with a more extensive root system can access a larger volume of soil, making it more resilient to drought stress and better able to find nutrients even in less fertile growing conditions. These improvements do not represent a fundamental change in the plant's genetic potential. What changes is the plant's ability to express its existing genetic capacity fully without being limited by biological bottlenecks in nutrient availability or root function. When roots encounter the diluted fermented mixture in the soil, a cascade of biochemical responses begins almost immediately at the cellular level. The organic acids produced during fermentation, primarily lactic acid and acetic acid, lower the pH in the immediate vicinity of root surfaces. This localized acidification helps dissolve mineral particles that would otherwise remain inert, releasing ions of calcium, magnesium, potassium, and trace elements into the soil solution where root hairs can absorb them. At the same time, living microorganisms introduced through the fermented liquid begin colonizing the rhizosphere. Plant roots constantly secrete sugars, amino acids, and other carbon compounds into the surrounding soil, and these secretions attract and feed beneficial bacteria and fungi. When microbial populations are robust, they form complex communities that interact with roots in ways that benefit both organisms. Some of the most important interactions involve plant hormones. Specific bacterial species produce auxins, cytokinins, and gibberellins as natural byproducts of their metabolism. These compounds, even in tiny concentrations, profoundly influence plant growth patterns. Auxins stimulate root elongation and branching. Cytokinins promote cell division in shoot tissues, and gibberellins regulate stem elongation and flowering timing. When beneficial bacteria are abundant in the root zone, plants receive a steady supply of these growth-regulating compounds. Inside the plant itself, improved nutrient availability triggers changes in metabolic efficiency. Chloroplasts function more effectively when magnesium supplies are adequate, because magnesium forms the central atom in every chlorophyll molecule. With more functional chlorophyll, the plant captures light energy more efficiently and converts it into sugars at a higher rate. Those sugars fuel all other growth processes, from root expansion to fruit development, creating a positive feedback loop where better nutrition leads to better photosynthesis, which generates more energy for further growth. Potassium plays a particularly crucial role in distributing the products of photosynthesis throughout the plant. It regulates the opening and closing of stomata and activates enzymes involved in sugar synthesis and transport. When potassium levels are optimal, sugars produced in leaves move efficiently through the phloem to wherever they are needed most. During flowering and fruiting, this means more energy reaches developing tomatoes, allowing them to grow larger and ripen more completely. The fermented mixture does not supply large quantities of any of these nutrients directly. What it does is optimize the biological and chemical conditions that allow plants to access nutrients already present in the soil, transforming potential fertility into actual plant-available nutrition. In soil that genuinely lacks essential minerals, no amount of microbial activity will create nutrients from nothing. But in most garden soils that have received organic amendments over time, the limiting factor is rarely total nutrient absence, but rather the biological capacity to mobilize those nutrients when plants need them most. The two tomato plants standing side by side in the same garden bed tell a story that has nothing to do with forcing growth or manipulating biology through artificial means. One plant struggles because the biological systems that should be delivering nutrients to its roots have weakened or disappeared entirely. The soil may contain adequate minerals, but without active microbial populations to dissolve those minerals and convert them into forms the plant can absorb, they remain locked and unavailable. Pale leaves, dropped blossoms, slow fruit development, and weak stems all reflect the same underlying problem: a breakdown in the biological partnership between roots and soil organisms. The other plant thrives because that partnership has been restored. The fermented mixture introduced living microorganisms and the organic compounds they produce directly into the root zone at the exact moment when the plant needed them most. As microbial populations multiplied and began processing organic matter in the soil, nutrients that had been unavailable suddenly became accessible. Root hairs expanded, chlorophyll production increased, and the metabolic machinery that powers flowering and fruit development began operating at full capacity. This difference reveals something important about how soil fertility actually works. For most of the history of agriculture, farmers understood instinctively that soil was alive. that it required feeding and care beyond simply adding mineral nutrients. They returned organic matter to the land, allowed fields to rest, and used fermented preparations to support crops during critical growth stages. These practices were not based on scientific understanding of microbiology, but they worked because they aligned with the biological processes that regulate nutrient cycling in natural ecosystems. Industrial agriculture moved away from this approach not because it was ineffective, but because it did not fit the economic and logistical requirements of large-scale mechanized farming. But home gardeners operate at a completely different scale, one where biological methods become practical again. A few liters of fermented liquid prepared in a kitchen container can treat an entire backyard garden, and the time investment is measured in hours rather than days. What makes this knowledge valuable is not that it replaces modern fertilizers or represents some superior alternative to conventional methods. Its value lies in addressing a specific problem that chemical inputs alone cannot solve: the gradual depletion of soil biology that occurs when living systems are managed purely through chemistry. Rebuilding microbial communities restores resilience, improves nutrient efficiency, and creates conditions where plants can thrive with less intervention rather than more. The method is simple, but the principle behind it is profound. Soil is not a chemical warehouse waiting to be restocked with synthetic nutrients. It is a living ecosystem where billions of organisms mediate the flow of energy and matter between minerals and roots. When that ecosystem functions well, plants respond in ways that no amount of fertilizer alone can replicate. The fermented mixture made from water, yeast, and sugar does not create this ecosystem from nothing. What it does is reawaken biological processes that were always supposed to be there, processes that tomatoes and their wild ancestors evolved alongside for thousands of generations. The response gardeners observe in their plants is not a trick or a shortcut. It is simply what happens when the biological foundation of soil fertility is restored, even partially, and plants can finally access the resources that were present all along, but remained out of reach. That simple jar of bubbling liquid sitting on a kitchen counter represents something larger than a gardening technique. It represents the possibility of working with natural systems rather than constantly trying to override them, of rebuilding rather than depleting, of understanding that the most effective solutions are often the ones that align with how living systems actually function. The knowledge was never truly lost. It survived in traditional farming communities, in organic gardening movements, and in the practices of growers who never stopped paying attention to what the soil was trying to tell them. Now it is returning to gardens where tomato plants are finally producing the harvest they were always capable of. Once the biological barriers were removed and the ancient partnership between roots and microorganisms was allowed to function again. Two plants, same soil, same sunlight, same water. The difference was never about adding something the plant did not need. It was about restoring something that should have been there all along.